Hi, I'm Dr. Lawrence Cooper. I'm at the MD Anderson Cancer Center, and I want to tell you a little bit more about what we're doing in our lab and how we're helping patients with cancer. With me today is David. He's an outstanding MD PhD student. David's getting not one, but 2 degrees. That's how brilliant he is. So I want to share with you some exciting data that Dave has generated around our ability to control T-cell growth after we've infused them into the patient. So David, tell us a little bit about what you're doing.
So our project is centered around ablating T cells that have been put into a patient in the event that there's a serious adverse event, that the patient is affected, that the T cells aren't doing what we want them to do.
So this is a type of safety switch?
Yes. The intention is to rapidly get rid of the cells that are inside the patient, so that there's no risk to the patient in getting this new type of T-cell therapy.
So T-cells being a type of immune cell.
Yes.
So how is it that you're putting the safety switch into the T-cell? How do you get that done?
So in the lab, we use chimeric antigen receptors, and they expand the T-cells by acting like the T-cell receptor that's naturally on the T-cell. And as they do that, the gene that codes for the chimeric antigen receptor can also code for this safety switch, which is icaspase 9, an inducible caspase 9.
So you're able to genetically modify a T cell to put in 2 genes? You can put in an immunoreceptor that can target tumors, and you can put in a safety switch?
Yeah, and we can...
And you're playing with people's DNA, their own T cells.
Yeah.
And you can get that to work.
Yeah.
So what are some of the data? Tell us what happens when you put these genetically modified T cells and you get rid of them with a special drug, or how do you get rid of them? What's the trick to get rid of them?
So amazingly, this has been done in patients already in a different kind of T cell setting, where they were giving T cells back to sick kids. And when the sick kids were getting transplant for their cancer, they got T cells back to prevent them from getting an infection. But sometimes T cells can attack the person's body when the T cells aren't from somebody.
That's graft versus host disease, right?
Graft versus host.
Yeah.
And they had to give a drug that activated this icaspase 9, and it killed the T cells and completely got rid of disease right after the drug was given. It was amazing results. And what we're hoping for icaspase 9 is that the same drug can be used to treat T cells used for a different reason, where the T cells are actually treating the ALL or the leukemia that the patient has, which we're targeting.
Sounds fascinating. Congrats on your data. So David is outstanding, as you can hear. And what he's really helping us understand is that the gene therapy platform we have developed to introduce the chimeric antigen receptor, in other words, to redirect the specificity of T cells to the patient's underlying tumor. That same gene transfer platform can be adapted to not just express the immunoreceptor, but also this icaspase, this inducible caspase 9. This is added safety for the patient. So we can give large numbers of tumor-specific T cells, and in the unlikely event something happens, we can get rid of those cells using a simple drug. Thanks very much.